The term designer baby often evokes images from science fiction. However, it refers to a baby whose genetic makeup has been intentionally selected or altered. This alteration typically aims to ensure the presence of specific desirable traits. It might also be used to eliminate particular defects from the child's genetic code. These modifications are usually achieved through genetic engineering techniques applied at the embryonic stage. The concept itself sparks considerable debate. It touches upon profound questions about humanity, ethics, and the future we are shaping with science. The technology is advancing rapidly. Therefore, understanding its implications is more crucial than ever. Genetic engineering, in a broader sense, is the direct manipulation of an organism's genes using biotechnology. It involves changing the DNA, the blueprint of life, within an organism. This can mean adding new genes, removing existing ones, or altering their function. For decades, scientists have used genetic engineering in agriculture and medicine. For instance, it helps create disease-resistant crops or produce vital medicines like insulin. Applying these powerful tools to human embryos, however, opens a new realm of possibilities and concerns. The precision and potential impact of human genetic engineering are unprecedented. This field requires careful consideration and public discourse. The journey into human genetic modification has been swift. Early discussions focused on theoretical possibilities. Now, we possess tools that make these possibilities increasingly tangible. The power to edit the human genome brings with it an immense responsibility. We stand at a crossroads, contemplating how to use this power wisely. The potential to alleviate suffering is enormous, yet the risks of misuse or unintended consequences cannot be ignored. This technology compels us to reflect on what it means to be human. It also forces us to consider the kind of society we wish to build for future generations. This essay will explore the multifaceted world of designer babies and genetic engineering. We will delve into the key technologies that make these interventions possible. We will examine the potential benefits, such as preventing devastating diseases. Crucially, we will also address the significant ethical concerns and potential societal impacts. The international legal frameworks governing this domain will be reviewed. Finally, we will discuss recent advancements and offer recommendations for regulation, particularly considering the unique challenges faced by countries like India. Our journey through this complex topic aims to provide a clear and balanced perspective. Germline gene editing is a central technology in the discussion of designer babies. This process involves modifying the DNA of an organism's reproductive cells. These cells are the sperm, eggs, or the embryo itself. The critical aspect of germline editing is that the changes made are heritable. This means they can be passed down to subsequent generations. This permanency is both a source of great potential and profound ethical worry. Due to safety concerns and complex social implications, germline gene editing for reproductive purposes is currently prohibited in over 40 countries. The debate continues about its appropriate uses. In vitro fertilization, commonly known as IVF, is another cornerstone technology. IVF is a medical procedure where an egg is fertilized by sperm outside the human body. This fertilization occurs in vitro, which means in glass, such as in a laboratory dish. Once fertilization is successful and an embryo develops, it is then transferred into a woman's uterus. The goal is to achieve a successful pregnancy. IVF was initially developed to help with infertility. However, it also provides a crucial window for genetic interventions. Embryos cultured in the lab can be screened or modified before implantation, making IVF a key component of many genetic engineering applications. CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, has revolutionized genetic engineering. It is a powerful and precise gene editing tool. CRISPR allows scientists to make specific changes to the DNA of cells with relative ease. It can be used to add, remove, or alter genetic material at particular locations in the genome. The discovery of CRISPR has dramatically accelerated research in genetics. It has opened up new avenues for understanding gene function and developing treatments for genetic diseases. Its efficiency and versatility have made it a popular choice for researchers worldwide. A specific and widely used CRISPR technique is CRISPR-Cas9. In this system, the Cas9 enzyme acts like a pair of molecular scissors. It is guided by a piece of RNA to a specific sequence in the DNA. Once at the target site, Cas9 cuts the DNA strand. This cut then allows scientists to remove a segment of DNA or insert a new one. This process can correct genetic defects or introduce desired traits. CRISPR-Cas9 is considered simpler, more versatile, and often more efficient than older gene editing tools. Its development has been a significant breakthrough, further propelling the field of genetic engineering forward. One of the most compelling arguments for genetic engineering is its potential to reduce the risk of genetic diseases. Many devastating conditions, such as Huntington's disease, cystic fibrosis, and Tay-Sachs, are caused by single-gene mutations. Germline gene editing could, in theory, correct these mutations in embryos. This would prevent the child from developing the disease. It could also stop the disease from being passed to future generations. The prospect of eradicating such inherited conditions offers immense hope. It could alleviate untold suffering for countless families across the globe. This potential alone drives much of the research. Beyond preventing specific monogenic disorders, genetic engineering could lower the probability of various inherited medical conditions. Many common diseases, like certain cancers, heart disease, and diabetes, have genetic predispositions. While these conditions are complex and influenced by multiple genes and environmental factors, identifying and modifying risk-associated genes could reduce an individual's susceptibility. This could lead to a healthier population overall. Imagine a future where the genetic lottery is less of a determinant for one's health. Such advancements could significantly extend healthy lifespans and improve the quality of life for many people. The ability to prevent or reduce the incidence of serious diseases translates directly into an improved quality of life. Individuals born free from debilitating genetic conditions would have the chance to live fuller, healthier lives. Families would be spared the emotional and financial burdens of caring for chronically ill loved ones. The societal benefits could also be substantial. A healthier population means reduced healthcare costs. It also means a more productive workforce and fewer resources diverted to managing preventable illnesses. This focus on alleviating human suffering is a primary motivator for many scientists in this field. Furthermore, the broader public health implications of genetic engineering are significant. By addressing genetic diseases at their root, we could see a dramatic decrease in their prevalence. This is akin to how vaccines have reduced the burden of infectious diseases. The cumulative effect could be a healthier global population. This would allow societies to allocate resources to other pressing health and social challenges. While the path to realizing these benefits is complex and fraught with ethical considerations, the promise of a healthier future remains a powerful driving force for continued research and careful development of these technologies. The conversation around genetic engineering often extends beyond disease prevention to include human enhancement. Some proponents suggest that gene editing could potentially lead to children with traits considered desirable, such as higher intellect or perhaps even greater obedience. The research notes mention this possibility, suggesting it might increase an individual's success in life. However, these claims are highly speculative and raise significant ethical questions. Intelligence and behavior are incredibly complex traits, influenced by many genes and environmental factors. The ability to precisely engineer such characteristics is far from certain. Moreover, the very idea of selecting for such traits is contentious. Another aspect of enhancement involves the potential to add new characteristics to future generations. This could involve conferring resistance to certain infectious diseases, as was the aim in the controversial Chinese experiment involving twin girls and HIV resistance. The scientists involved claim to have modified a gene called CCR5 to make the babies less susceptible to HIV infection. While the health of these children is reportedly normal, the experiment drew widespread condemnation for ethical breaches. The idea of adding traits that could benefit society, such as enhanced immunity or metabolic efficiency, is theoretically appealing to some, but it also opens a Pandora's box of unforeseen consequences and ethical dilemmas. It is crucial to differentiate clearly between therapeutic uses of gene editing and enhancement applications. Therapeutic uses aim to correct genetic defects and treat or prevent diseases. This is generally viewed more favorably by the public and the scientific community. Enhancement, on the other hand, aims to improve upon what is considered normal human capability or appearance. This area is fraught with ethical concerns about fairness, equality, and the potential for creating social stratification. The line between therapy and enhancement can sometimes be blurry, making robust ethical guidelines and public discussion essential. Defining these boundaries is one of the greatest challenges we face. The case of Adam Nash is often cited in discussions about designer babies. Adam was conceived through IVF and pre-implantation genetic diagnosis, PGD. This was done to ensure he was a suitable tissue match for his older sister, who suffered from Fanconi anemia. He was not genetically engineered for enhancement. Instead, PGD was used to select an embryo that was both free of the disease and an HLA match for his sibling. This allowed him to be a donor for a life-saving bone marrow transplant. This case highlights the use of genetic technologies for therapeutic benefit within a family, distinct from germline editing for enhancement. Section 5. Navigating the labyrinth, ethical dilemmas, and societal concerns. One of the immediate ethical concerns associated with manipulating embryos is the risk of procedural failure. Gene editing techniques, while increasingly precise, are not infallible. There is a possibility that the editing process could go wrong, leading to unintended genetic changes or damage to the embryo. In such cases, the embryo might not be viable, or it might develop with unforeseen health problems. This raises the question of what happens to modified embryos deemed unsuitable for implantation. The potential for embryo termination as a consequence of failed genetic procedures is a significant moral consideration for many people. It underscores the responsibility involved in these interventions. A major societal concern is the potential for genetic engineering to create or exacerbate social inequalities. If access to these technologies is limited by cost, only the wealthy might be able to afford genetic enhancements for their children. This could lead to a genetic divide, creating a society with biologically enhanced haves and unenhanced have-nots. Such a scenario could deepen existing social stratification and lead to new forms of discrimination. The prospect of humans with perfect traits, as suggested in some discussions, raises fears of a dystopian future where natural human variation is devalued. Ensuring equitable access and preventing genetic elitism are critical challenges. The use of germline gene editing also raises profound questions about individual autonomy and choice. Children born with genetically modified traits will have had no say in these alterations to their fundamental biology. These changes are permanent and will be passed on to their own offspring. This limits their autonomy over their own genetic makeup and future. Some ethicists argue that every individual has a right to an open future, free from predetermined genetic modifications that might constrain their life choices or sense of identity. The idea of parents designing their children like products also raises concerns about the commodification of human life. Furthermore, there are concerns about the long-term impact on the human gene pool. Germline modifications are heritable, meaning they become a permanent part of the human genetic legacy. Introducing widespread genetic changes could have unpredictable and potentially irreversible consequences for human evolution. There is a risk of damaging the gene pool if modified traits lead to unforeseen health problems in later generations or reduce genetic diversity, which is crucial for adaptation. The high cost of these technologies also makes them inaccessible to most of the global population, further compounding concerns about fairness and the responsible stewardship of human genetics. Section 6. Global Governance, International Legal and Regulatory Landscapes. The governance of human genetic engineering, particularly germline editing, varies significantly across the globe. There is no single universally accepted international treaty specifically regulating all aspects of this rapidly evolving field. However, a prevailing sentiment, reflected in the laws of many nations, is one of caution. Over 40 countries have implemented prohibitions or strict regulations on germline gene editing for reproductive purposes. These restrictions are typically based on concerns about safety, ethical implications, and the potential societal impact. The international community continues to grapple with how to best manage these powerful technologies. In the United States, the regulatory landscape is complex and somewhat fragmented. There are no specific federal laws that comprehensively ban or permit human germline modification. However, the National Institutes of Health, NIH, has consistently stated that it will not fund research involving the editing of human embryos for reproductive purposes. In 2016, NIH guidelines affirmed this stance. A subsequent report from the National Academies of Sciences, Engineering, and Medicine advised the government on potential benefits and outlined stringent conditions under which clinical trials might one day be considered. The Food and Drug Administration, FDA, has regulatory authority over clinical applications, and it has allowed the use of embryos for research related to infertility issues. Research proceeds cautiously under these guidelines. The United Kingdom has adopted a more structured and arguably more permissive regulatory approach compared to the US. The Human Fertilization and Embryology Act of 1990 and its subsequent amendments established the Human Fertilization and Embryology Authority, HFEA. The HFEA licenses and monitors research involving human embryos. While initially restrictive, the UK has shown a willingness to adapt its regulations. For instance, in 2015, the UK became the first country to legally approve mitochondrial replacement therapy, a technique to prevent the transmission of mitochondrial diseases. The HFEA has also granted licenses for research involving the genetic modification of human embryos, though not for implantation. Other major nations are also developing their regulatory frameworks. China, following the highly publicized and controversial experiment by He Jiankui that resulted in the birth of genetically edited twin girls, has moved to strengthen its oversight. The government is in the process of introducing more comprehensive gene editing regulation, and revisions to its civil code now include provisions specifically addressing gene editing. Japan currently lacks a specific legal framework for genome editing in humans for reproduction. However, a 2015 report by a government advisory group accepted gene editing research for basic science, but opposed the reproductive manipulation of embryos and the implantation of modified embryos. Japan is reportedly moving towards enacting a more formal regulatory law. Section 7. Ownership of Life's Code, Intellectual Property Rights in Gene Editing. The rapid advancements in genetic engineering have given rise to complex questions regarding intellectual property rights. A central debate revolves around whether genes, or the tools to modify them, can be patented. Patents grant inventors exclusive rights to their inventions for a certain period, which can incentivize research and development. However, when applied to genetic material, patenting raises ethical and accessibility concerns. The ability to patent human genes or gene editing technologies like CRISPR has been a subject of intense legal battles and public discourse. Different jurisdictions have adopted varying approaches to this intricate issue. In the United States, a landmark Supreme Court case significantly shaped the landscape of gene patenting. In 2013, the court unanimously ruled that naturally occurring DNA segments are products of nature and therefore cannot be patented. This decision invalidated myriad genetics patents on the BRCA1 and BRCA2 genes, which are associated with increased risk of breast and ovarian cancer. However, the court did allow for the patenting of complementary DNA, cDNA, which is a synthetic form of DNA, as it is not naturally occurring. This ruling has had a profound impact on genetic research and diagnostic testing in the US. The European Union has a somewhat different stance on gene patenting. The EU's Directive on the Legal Protection of Biotechnological Inventions, 98/44/EC, allows for the patenting of an element isolated from the human body or otherwise produced by means of a technical process, even if the structure of that element is identical to that of a natural element. This means that a gene, once isolated and its function identified, can potentially be patented if it has a specific industrial application. This approach aims to encourage innovation in biotechnology while still acknowledging that the human body itself is not patentable. The nuances in interpretation continue to be debated within member states. India's approach to patenting genetic material is guided by the Indian Patent Act, 1970. Section 3(c) of the Act states that the mere discovery of a scientific principle or the formulation of an abstract theory or discovery of any living thing or non-living substance occurring in nature is not patentable. Furthermore, Section 3(j) excludes plants and animals in whole or any part thereof other than microorganisms, but including seeds, varieties and species, and essentially biological processes for production or propagation of plants and animals from patentability. Gene modifications are also argued to be unpatentable on ethical grounds due to their potential impact on humanity and the environment. This reflects a more restrictive stance on patenting life forms and naturally occurring substances. Section 8. The cutting edge, recent advancements, and future directions. The field of genetic engineering is characterized by rapid and continuous innovation. Recent advancements, particularly with tools like CRISPR-Cas9, are pushing the boundaries of what is possible. Scientists are increasingly using these molecular scissors not just for basic research, but also for developing novel therapies. For example, CRISPR-based therapies are being explored and in some cases, clinically trialed for treating diseases such as sickle cell anemia, beta thalassemia, and certain types of cancer, including leukemia. The ability to create designer immune cells, genetically engineered to better target and destroy cancer cells, represents a significant step forward in oncology. Another area of active research involves refining the delivery and precision of gene editing tools. One approach being investigated is the direct injection of CRISPR components into eggs or very early embryos. The aim is to perform genetic modifications at the earliest possible stage, potentially increasing efficiency and reducing the risk of off-target edits, unintended changes at other locations in the genome. While this research is primarily conducted in animal models or on human embryos not intended for implantation, it highlights the ongoing efforts to improve the safety and efficacy of gene editing technologies. These technical refinements are crucial for any potential future therapeutic applications in humans. The concept of gene doping has also emerged, raising ethical concerns, particularly in the realm of sports. Gene doping refers to the non-therapeutic use of gene therapy to enhance athletic performance. This could involve modifying genes to increase muscle mass, endurance, or oxygen transport. While still largely theoretical and unproven in humans, the potential for gene doping is taken seriously by anti-doping agencies. It represents a misuse of powerful medical technologies for non-medical enhancement, blurring the lines between therapy and performance augmentation, and posing risks to athletes' health and the integrity of competitive sports. Pre-implantation genetic diagnosis, PGD, is an established technology that often intersects with discussions of designer babies. PGD involves the genetic screening of embryos created through IVF before they are implanted in the uterus. Its primary use is to screen for specific genetic diseases or chromosomal abnormalities, allowing parents to select embryos that are unaffected by a particular condition. While PGD itself does not involve altering genes, it allows for the selection of embryos based on their genetic makeup. This technology is widely used and accepted for preventing serious genetic diseases, but debates arise when considering its use for selecting non-medical traits. Section 9. Charting the Path Forward. Regulation, Risks, and India's Unique Challenges. As genetic engineering technologies continue to advance, the need for robust and thoughtful regulation becomes increasingly urgent. A global consensus is emerging that procedures involving human embryos, particularly heritable germline modifications, should require explicit authorization from a designated central government body or relevant competent authority. This oversight is essential to ensure that any such interventions are conducted ethically, safely, and with societal consent. Establishing clear lines of authority and accountability is a fundamental step in responsible governance of these powerful tools. Such oversight must be transparent and involve broad societal input. Further regulatory measures should include a mandatory licensing system for any research or clinical practice involving gene editing on human gametes or embryos. Laboratories and clinics conducting such work must be subject to stringent controls and regular inspections by government authorities. This would help ensure compliance with established protocols, safety standards, and ethical guidelines. Moreover, given the profound implications for human life and future generations, any non-compliant acts or unauthorized uses of these technologies should be considered serious, cognizable offenses, carrying significant legal consequences. Public trust depends on rigorous enforcement of agreed-upon rules. India faces a complex situation regarding the regulation of genetic engineering. Currently, there is no specific overarching law that directly regulates genetic modification in humans or germline gene editing. The Indian Council of Medical Research, ICMR, has issued guidelines that deem the creation of designer babies for reproductive purposes unethical and recommend its prohibition due to insufficient knowledge about safety and long-term effects. However, the legal enforceability of these ICMR guidelines is a subject of debate, as they are not legislated statutes. The Delhi High Court, in one case, observed that government-issued guiding principles do possess legal validity until they are inconsistent with existing laws and should be adhered to ethically. This leaves a degree of ambiguity. India's unique socio-cultural context presents specific challenges and risks related to designer baby technologies. The country has a well-documented history of prenatal sex determination and female feticide, driven by a strong societal preference for male children. The advent of technologies that could allow for the selection of traits, including sex, before implantation, raises serious concerns that it could exacerbate the already skewed sex ratio and further devalue female children. Therefore, any regulatory framework in India must be acutely sensitive to these potential misuses. It must include strict prohibitions on sex selection for non-medical reasons and robust enforcement mechanisms to prevent the misuse of these powerful genetic tools, protecting the vulnerable and upholding ethical principles. The journey ahead requires careful navigation, balancing scientific progress with profound ethical responsibilities. Public engagement, transparent governance, and international cooperation will be essential to ensure that genetic engineering technologies are used for the benefit of all humanity, respecting human dignity and promoting a just and equitable future.